Okay,
Oh, there you go. It's recording. That's good. Thank you.
okay,
Um,
it's recording now.
okay, fab. So let me just find my questions for you. Um, so apologies, I know you've sort of talked me through some of these things already, but I was just wondering if you could sort of very much in layman's terms explain the work you've been doing in 2020. I know it's continuation from earlier work, but yeah, just how you'd explain it to a small child, basically.
Oh, okay. Um, or my parents. Um, okay, so I don't actually go to Antarctica. There's a whole set of people who are drillers who drill the ice cores. So that's set up many years in advance, and so they set um, spend several years planning and then building the equipment primarily in the U.S. For the U.S. Expeditions anyway, and then they haul it all down to Antarctica to the site and set it up. That also takes several months to set all of that up, including the housing and everything in Antarctica, because a lot of the sites are very remote for the drill. They pick the sites where they'll get the best ice cores, meaning the right thickness, the right layering, the right time scale, because as you drill down, you go back in time essentially. In fact, I think I can't remember the guy's name, but he wrote a book called The 2 Mile Time Machine, and it was talking about the ice core. Oh, it's Richard Alley at Penn State. Um, so they have to drill down, and they have an ice coring drill that is about I don't know 20 feet. That would be like 6 or 7 meters long, where the top half is all the mechanics and motors and everything, and the bottom half collects the core inside of a tube. And so they bring it up about 1 or 2 meters at a time. And so the deeper you go, the longer it takes to go down and come back up with the core. And the cores are then brought up, archived there, and they actually split them so that every other core stays there in Antarctica, and all the other alternate cores are shipped back to whatever site they happen to be in. In the U.S., it's primarily Denver, Colorado. So that's where we begin. Actually, we've gone to Denver, Colorado, to the Ice Core Laboratory, the NSF Ice Core Laboratory, to pick up cores, the ones that we want. We've already studied what amount of time we're interested in, what particular cores. For Lake Vostok, we were interested in what's called the accretion ice. And so as this 2 mile thick, 3,700 meter glacier moves over the lake, and the lake is as big as Lake Ontario and Lake Erie. So it's a very, very large lake. It's, I think, the sixth largest lake in the world, um, by volume. It's actually deeper than Lake Erie and deeper than Lake Ontario. So it's a huge lake, but it's under all that ice. And as it goes over, some of the water from Lake Vostok freezes to the bottom of the glacier, and that's called the accretion ice. So as it moves across, it accretes to about 200 meters or 210 meters thick on the far side, which is where the drill site is. And so it's effectively moved across the lake, collecting little samples of surface water all the way across. And so it also contains a timeline of the lake, which is about I think around 15,000 years as it moves across. So you have two different timelines. The the Vostok ice core goes from zero to about at least 4 or 500,000 years. Or yeah. So, and at the bottom, it's kind of crumbled because it hits the base, the rock on the at the bottom of the glacier, the bedrock. and it's all jumbled. But some estimates are that the ice there is between 1.5 million years old. So there's some very old ice. And then you hit the accretion ice, where the closer it is to the glacier, the older it is, and as you go down, it's actually younger. So it's sort of the inverse of the glacial ice. So anyway, we were interested in the particularly both the basal ice to see what is going into the lake from the glacier, because some of the glacial ice melts as it goes into the lake. The lake is a little warmer than freezing at that pressure. And also the accretion ice from the lake to sample the lake water. So we used different methods, two different methods mainly. One was to culture organisms from that ice. And I can't remember how many, we have several thousand cultures now growing in our lab. So these are living organisms, primarily bacteria and fungi, that we've grown in our lab from that accretion ice. It has very low levels of organisms in it. As you might expect, it's what's called oligotrophic, which means the lake water has very, very few nutrients in it for these organisms to live on. Although the lake seems to have a hydrothermal vent somewhere in it, probably on the south west corner. It looks like there's at least hydrothermal activity there, which provides the energy for these organisms to live. So as far as organisms, pretty much about half of them we can identify, and the other half we don't know exactly what they are. So there are some unknown organisms. We can kind of place them compared to other sequences, because we actually, after we grow them, we do DNA sequencing on them to try to get an identity for them. And so that's one part. That's a slow process. And so when I have graduate students in my lab, they want to finish in a reasonable amount of time. A lot of these organisms, it takes 6 months just to grow them. And sometimes the students would culture 100, 200, 300, 400 plates, put this melt water on the plate, and they might get one culture from all of that. And so, I got a lot of students with worried faces on there, especially early on when not much would grow. I should mention that before we melt the ice, we sterilize the surfaces of the ice so that there are no contaminants on it. So we have an elaborate process in a sterile room to both sterilize the outside of the ice surfaces, and melt the ice in a sterile environment. So we're fairly certain that we're getting rid of almost all the contamination. We also run controls on things that we take through the same process, what we call sham ice cores that we make in the lab. So that any organism that we get in both places, we subtract those from the final results, just to be sure that we're not picking up any contaminants. Okay, so that's the culture work. We also, to speed things up, do what's called metagenomics and metatranscriptomics, and that's where we're trying to sequence all of the DNA and RNA in the sample. And so that's a different process, but what it does, it will give us what DNAs are in that water. They're not necessarily all living, so these are just sequences that are floating around and happen to get frozen into the ice. So with the culture work, we're getting several thousand cultures over a 10-year period, essentially, 10 or 15-year period. With the metagenomic results, we can pick up uh, thousands of sequences just in one experiment. And so and that's, takes about, I don't know, 3 months to do all of that. So in 3 months, you can actually have more information than it took us 10 years to do before. So it speeded things up quite a bit. So from that, then we take the meltwater, we use a process called reverse transcription, which synthesizes a DNA copy of the RNA. We also use the DNA that's in the sample. So we're looking at both the DNA and the RNA in the sample. And then go through a process called, polymerase chain reaction, or PCR. A lot of people have heard of that. Amplify these so that then we can get enough of those nucleic acids to sequence, and then with next generation sequencing methods, we obtain the sequences, compare those to known sequences on national and international databases, and then try to come up with identities for all of those organisms represented by the sequences. And so in doing that, we've looked at essentially from glacial ice above Lake Vostok, from the basal ice that's interacting with the bedrock and feeding into Lake Vostok, and several locations within Lake Vostok, which is, as I said, the southwest corner where there's a bay. And in the bay, it looks like that's where the hydrothermal vent is. And then further out, you go into a deep basin, the main basin of the lake. And so we've sampled each one of those areas, and we find very different organisms going into the lake than we find in the lake itself. There's some overlap, but by and large, they're different organisms, indicating that there's a community of organisms living at the base of the glacier. And then there's a different set of organisms growing in the lake, and they differ in that hydrothermal vent area from the main basin. So there are crossovers between all of those. I mean, there are common species in each one of those, but they're pretty different. That was our indication that yes, things are living in the lake, and there are at least 1,500 to possibly over 3,000 different species, primarily bacteria, that are living in the lake. So it's running about 95% bacteria and 5% eukaryotes, and the main eukaryotes are fungi. But then we find some larger organisms, including small animals like rotifers and copepods, which feed mainly on algae and bacteria, or on each other. And
just really quickly, Scott, sorry to interrupt.
Yep, sure.
So there's the bacteria and the fungi, and you just mentioned some other creatures. Are they fish, or what are they?
No, they're small crustaceans.
Okay.
So yeah, we do so in our initial studies that came out in 2013, we didn't find any sequences from fish. We found like anemones, like hydra and things like that. So some very simple animals, small animals, but didn't find any fish. But we did find several bacteria that only are known to grow in the intestines of fish. So that indicates that, the fish are defecating in the water, and these bacteria are released that way. And so that's probably how they were winding up in the ice. But in our more recent study that we just published last year, we found an authentic fish sequence, and the thing that it's closest to is an Antarctic rock cod, which they're small cod that live essentially all the way around coastal Antarctica. Uh, so, that would I don't that was pretty convincing to me that now we finally have found the fish. If the fish are growing down there, then they're probably very small because there aren't a lot of nutrients down there, but there's enough coming from well, the heat from the thermal vent, and it's releasing minerals and things like that. And there are bacteria then, and also, um, well, mainly bacteria that can fix carbon down without any sunlight. So there are a lot of organisms that are um, the type of bacteria that can still fix carbon, so you can get nutrients that way. And the higher organisms then can feed off of those and get their carbon compounds from that. So we've done some studies on nitrogen cycling and carbon cycling, and there's enough there to have a complete community down there that would survive. I mean, that was one of the original criticisms, that nothing can survive down there, right? It's high pressure, it's cold, there's no light. And at that time, they didn't think there was any energy source, but now we know that there is at least one energy source, and that's the hydrothermal vent or ridge. Some people say it's an entire ridge. Um, and enough nutrients down there being fed in by the glacier, because all that snowpack is eventually melting. Part of it is melting into the lake, and that's bringing not many nutrients, but it's actually bringing a lot of oxygen and nitrogen into the system uh, due to the trapped atmospheric air from the snow above, and it gets packed into the ice, and eventually some of that is released. So there are different inputs into that lake, definitely.
Just unmute there. Fantastic. And so the results when you found out that there could be life in the lake, was that
Mhm.
something you found out in 2013, or was that something you found out in 2020?
Well, our first indication was in 2008 and 2009 when we were
Yeah.
actually culturing things from there. So that told us not only was there life, but it was still living, that we did pick up living organisms. In 2013, those two papers that expanded the number of organisms that we found, because when you culture things, you can only culture about 1% of what's there.
Mm.
The other things, you don't, they might depend on other organisms to live, or they're just, we can't choose, you can't, if you don't know what's there, you don't know what culture media to grow them on, or what temperature, whether they need light or whether light inhibits them. So you can really only culture about, 0.1 to 1% of what's actually there. So that gave us an indication, at least, that something was living. The study in 2013, we didn't know what organisms were living, but we found, essentially 3,000 different species of organisms in there that expanded our view of what probably is there. And then in 2020, that's when we expanded it not only to the lake, but also to the basal ice and the glacial ice to find out where these organisms are coming from, indicating that now the organisms that are in the lake are different from the basal ice organisms. And so definitely the organisms that we found in the lake are not simply those that are melting from the glacier above, but they're resident organisms in the lake that are living there. So sort of three pieces to the puzzle.
Sorry, I was muted again. Um, Great. Okay. And so this 2020 research, would you say, or could you say this uh this 2020 research takes us a step closer to kind of finding out if there's life on other planets or the moons that we were discussing?
Well, to me, it just expanded my view of where things could live. So, and probably for others. I mean, when I was going through school, you, there was this Goldilocks zone. The Earth was in the Goldilocks zone, right? So only organisms could live there. There was no chance of finding anything on Mars or Venus or any of those places. Now that organisms have been found in hot pools, and they've been found um, deep in ice, still living, sometimes for, trapped in ice for 8 million years, and they can grow again. And now Lake Vostok presents another piece to that because it's under high pressure because of that 2 miles of ice on top of it. So now you've got no light, you have cold temperatures, you have high pressure, and it's oligotrophic, meaning that it has very few nutrients. That sort of expands our thoughts on where things can actually live. And so now, yeah, there are several possibilities in the solar system of finding life, and there's that Mars mission going on right now with Perseverance, and they're I think they're going to drill into the soil to find out how much water is in the soil. They know that there is, I mean, a long time ago, people thought that there was only carbon dioxide ice at the poles, and there is a cover of carbon dioxide ice. But underneath that ice is a layer of frozen water. And so there's water there. There's water on several of the moons of Jupiter and Saturn, as well as on, all the way out to Pluto and Charon. So there's a lot of ice around even in the solar system, more than I ever thought. And so there's a possibility in several of those cases. They're not me, but the astrobiologists are fairly certain that there are oceans of water under the ice. And in some cases on Enceladus, that's a moon of Saturn, and on at least on one of the other moons, there are thick layers of ice. And on Enceladus, they've also seen geysers that probably represent, I mean, the robots that went out there and took pictures, probably is crystals of ice that are being forced out first as water, but as soon as they hit the outside, they freeze. So there are these long jets going out, indicating a lot of um, um, activity, at least, probably because they're interacting with the gravity of the planet, and they're being moved around quite a bit, a lot of energy that way. The cracks in, it's not Titan, but the moon that you mentioned in your email, I can't remember right now. So
Is it Europa?
yeah, Europa. So that's another one. Um, and there are several others actually that have features on their surface that are, it's just indicating that that's ice. And I think they've done spectral analysis also, and it's mainly ice, and it's all crumbled, and a lot of movement of that ice. So underneath that indicates that there's something going on, maybe tides or something with the oceans below that are moving that ice around. But it's miles thick, so it's not just a few hundred meters or even 1 mile thick. Some of the ice appears to be um, 50, 100 miles thick. And so getting down into the oceans is going to be a challenge, to say the least, if they want to. I think, primarily, especially with the moons where the geysers are spewing, then you're getting probably water from those oceans or lakes below, and you could sample the surface of those moons to see what's coming out of that and whether it has any kind of life in it. But from the Murchison meteorites and from other things that we know, there are amino acids, there are precursors to nucleic acids, there are many other compounds that exist in meteorites that hit the Earth that have been sampled. So there are precursors to biological life that are being imported and exported from different locations in the solar system all the time. So there's a possibility that may be how life started on Earth. Most of the things were already here. Well, many of the things were already here. And it just took time and the right conditions to put those together into, what we call life now, living organisms with a membrane and amino acids and DNA and RNA and all that.
Fantastic, thank you. Let me see if I've got any other questions here. Oh yeah, so the samples that you've been looking at in 2020, do you know when they were drilled?
That's a good question. I think they have. I think they were drilled in the 1990s, but
Oh, really? That long ago?
yeah, I'd have to look into the dates. We obtained most of those ice cores when I was still in New York. So that was probably, let's see, 96, 97 was when we first obtained those cores, and then they've been in our freezers ever since then. We picked up a few other ice cores in a different project, some of the accretion ice in 2000. That's when I was here. Probably 2001, 2002, 2003, somewhere around there.
Okay,
Yeah.
and how exactly would you describe an ice core? Like, what exactly is it? Just kind of compacted ice? Is it something more complicated than that?
Yeah, so ice isn't just ice. There are many types of ice, right? So if you look through some of that stuff that I sent you in the book, it describes how a glacial ice is produced. And so you have snow falling on a surface, but it could be the glacier. And at first, there's a lot of air. It's 90% air and about 10% ice or snow. That the next year, it gets covered with more snow that becomes more and more compacted, until it's physically separate from the atmosphere. Then, it's sealed off. And that takes I think, about 100 meters of compaction from the snow down to where you get the real solid glacial ice down below. So when they drill down through what's called the firn, which is that upper layer that's still in contact with the atmosphere to some extent. And we don't use that because that can contain contaminant from the surface. And so we start about 100 meters down. And the ice cores, depending on the drill, because there are different drills, there are hand drills, there are solar powered drills, there are diesel powered drills. And the biggest drills have, the teeth are around the outside of the drill. And as I said, it's about, I don't know, 6 or 7 meters long. Just with that last 2 meters being where the core is gathered. And the largest ones are collect a core that I think is 10 centimeters across. And so each time they go down and come back up, you pick up what's called a core section. the whole core represents all the way from the top to the bottom. But then they're gathered in sections, and when we use them, we talk about a particular section, and we can relate that not only to the depth, but also the time. And in different places, the snow will collect at different rates. So at Vostok's a high desert, so it receives less than an inch of snow every year for the whole year. And so you can essentially go back further in time with a shorter core, go back, 500,000 years, whereas at some of the Greenland sites, the same thickness, if you go down, I think the one is like 1,500 meters, it's only about 100,000 years old, because the snow, you get more snow every year. So the ice cores are just simply, representations of the snow accumulation over the years. And at first you can, in some ice cores, at first you can see definite layers, but as it becomes more and more compacted, you can't see the layers anymore, so that you can't count back. And so the way that they date ice cores is by looking at radioactive elements that are in the ice. And so, and I can't remember one is a radioactive oxygen species, and I can't remember what the other ones are. I'm not a chemist, so I don't deal with that. I mean, many people use these ice cores so that you have chemists looking at them, you have geologists looking at them, you have climate scientists looking at them, and then a little small slice for us biologists. We get a little bit.
Okay, sorry. Just typing away as you speak.
Yeah, that's fine.
Lovely, apologist. Cool. Just looking at my questions again. So some of the stuff that's taken sort of 10 to 15 years to get results from, is that something you started 15 years ago? Or is that something you started in the 90s, so the results would have been sooner?
No, I mean, most of it, some of it's organized and some of it's not as organized. It depends. So I get students coming in, and I, give them the possibilities. I don't only work on ice, I work on introns and I work on other things. So the students come in and I present different projects that they could do. And especially on this type of project where contamination is it's important to make sure that students realize it's really critical to make sure that there's no contamination problem. I have to choose students specifically that can work under those conditions, or those understandings, and that they're very interested in the project, because a lot of times especially with the culturing, it was a tedious process. And so they have to, unless they go in really excited about it, that within a year they will become kind of less interested, I'll say, in continuing. So and that slows things down quite a bit, because if a student quits, then you have to find another student to sort of take up where that student left off. So a lot of it was, those sort of considerations getting things started. The 2020 work, I think Colby Gura started in, well, it only took him about 2 years, and that was an extension of what we'd already done. So that 2020 was pretty much adding on one more ice core in the accretion ice from the lake, or maybe two, and one from the basal ice, and then one from the glacier. So that actually all the ice for that project had already been melted, and so that was done by I think Tom and Rom about 10 years before that, and then we store the melt water for other students to use, because melting the ice, decontaminating the ice, melting it takes quite a while to do that also. So there were several pieces to that project. So it comes in stages. I'd say to study any particular ice core, to do the metagenomics, although it takes about 6 months to get the data, it takes another 6 months to analyze the data. And so altogether, for any particular ice core, for one or two ice cores, it would take a student 2 to 3 years to work on that.
Okay. And what would you say is your kind of overriding question that you want to answer? Like, why are you doing this, would you say?
Well, I'm interested in science overall, but I was just interested at first. Well, the way I got into the project originally, into working with ice cores at all, because I hadn't been working with that at all. I was working with just ancient DNA, ancient samples, starting back in 1985, when no one was doing that. And then I so I got my PhD, moved on to State University of New York in Syracuse. And there, Tom Starmer and Kathy Katranis, they were working on isolating these organisms from the ice cores. And at first, I didn't get into the project because I was working on other things. But eventually I had some time, and they kept asking me if I could do some work on that. So I started probably in 91, working with isolating organisms from this glacial ice, and then I kind of got hooked into how far back can you go, and I was interested in molecular evolution. So I thought we could do a time course of looking at mutation over real time in the ice, and you can't really do that with DNA organisms, but you could do it with RNA organisms. So that I got connected up with a virologist, and so we started looking at these viral sequences in the ice, and we did find a time course. We could look at mutation rates in those viruses over a 140,000 year time period. But they're also mixing in with that. It appeared that one of the ancient genotypes appeared more in more recent times, and they were sort of all mixed up. So we developed this idea of genome recycling, where the ancient organisms, melt out of the ice. They're still living, they grow, and so they're introducing ancient genes into the modern population. And for viruses, that turns out to be a good thing for viruses, because they can sit out in a glacier for a long period of time until their hosts no longer have immunity, and then they come out and they can start infecting the naive host. So that's how it started. And then I was able to get some cores from Lake Vostok, from the accretion ice, and that's when the story started to develop about Lake Vostok. And everyone was saying there's nothing living there, And so we tried to see, well, can we isolate any organisms from that? And we could. So that sort of started our journey into Lake Vostok. And then that just step by step developed into what's in Lake Vostok, in which parts of Lake Vostok are there more organisms, and then the recent study, what's coming into Lake Vostok um, versus what's actually in Lake Vostok, and that's what the 2020 was tying up. So, and I think we're done on Lake Vostok then, because I'm probably going to retire next year, and it's like, okay, I've probably done enough on that.
Fabulous. Okay, wonderful. And just quickly, because I've realised I've got a 3 o'clock chat as well, and I'm taking up a lot
Yep.
of your time. But is there one question that I was asking you, which you rightly said is preposterous, is if there's any viruses down there. Do you know if that was
Yeah.
actually a genuine concern at one point? Was it something anyone studied, as far as you know?
Only I think we're the only ones. I have to think hard, but I think we're the only ones that have been studying viruses in the ice.
Mm.
So, in a different study, we studied influenza A viruses for a while,
Mm.
and found them in. We got some ice samples from lakes in Siberia, in northeastern Russia, and showed that where you had a lot of birds, especially nesting birds, nesting geese and ducks, there was a lot of influenza frozen in the ice during the winter when the geese were no longer there. So subsequently, Danny Shoham from Israel showed that the viruses in the ice are actually living. And so again, we have this genome recycling, but in a pathogenic organism where the the geese deposit the viruses during the spring and summer when they're there in the water, and then they start flying south for the winter. The ice freezes, and there's still some viruses left in the lake, and they get frozen into the ice, as well as they've found them in the mud in those lakes. Well, not those lakes, but other lakes in Alaska. And the viruses are still living, and they can overwinter. And then the next birds to come in there, it's usually a different set of birds because they have different patterns of migration. And now they're exposed to the virus that the last birds leaving left the year before. And so you have this whole, I call it the lakes are mixing bowls for these viruses, and the birds are mixing bowls for the viruses in a different way. So you have a biological component and a non-biological component spreading those viruses around. And it probably is true of other viruses as well. There have been cases where anthrax has been frozen in the permafrost up in, I think that was also in Russia. And then all of a sudden, and that was like 75 years ago. And then recently, as the permafrost has been melting, there was a case where I think hundreds of reindeer were dying of anthrax.
Wow.
And a few people also, one person died because they also consume the meat and are in contact with the reindeer. And so there was an outbreak of anthrax, and they sort of traced it back to this melting permafrost where some of the old animals that had died 75 years ago were becoming exposed at the surface. And that may have been the cause of that infection or that epidemic.
Okay, lovely. Well, it's not lovely, but so, and what do you think? Could there be viruses in Lake Vostok?
I'm sure there are viruses down there. I'm not sure they are, that humans can contract them, but probably, fish and fungi and things like that are, and probably the bacterial virus. Viruses are actually in aquatic systems and marine systems. They turn out to be extremely important to nutrient cycling because they kill some of the organisms that releases all those nutrients from those organisms, and other organisms can actually feed on those. So as far as nutrient cycling, aside from any, pathogenicity, they turn out to be incredibly important in ecosystems. So a lot of times I focus on that part rather than the negative, rather than the negative parts of getting sick. Although with COVID, it's hard to ignore those sorts of things.
Yeah. Okay, lovely. And just quickly, are there any other mysteries that you're aware of that are being studied in Lake Vostok by kind of other scientists, like anything in the last kind of couple of years?
Not with Lake Vostok. They have been tapping into other lakes. They tapped into Lake Whillans a few years ago and took a probe all the way into the sediments as well. That lake is much shallower, so I think I think rather than being thousands of meters beneath the ice, it's only tens of meters, and it's a shallow lake. So they tapped into that a few years ago. Found actually some similar organisms that we have found in Lake Vostok. And then there are other people who are sending probes down under the ice, under sea ice, and in bays, looking at organisms down there. I think I sent you a video of one of those or two of those. Yeah, the work on Vostok kind of stopped a few years ago when the Russians penetrated into the water and then allowed the water to go up into the borehole. It froze, and the next season they went down and collected some of that water, but it was heavily contaminated all the way through with drilling fluid, and so it was essentially contaminated. They tried another year after that, and I think it had the same result. And they were years ago going to send a robot probe into the lake, but there was a lot of opposition to that because you would contaminate the lake and it would be hard to study after that. So that's been, I think, 2015, 2016, somewhere around there was the last I heard that anything was going on down there. And I think the funding for some of that kind of dried up. It costs a lot. It costs a lot to go down there and drill. So.
That's a shame that the samples are basically useless.
Yeah, they were, I think, very disappointed. But it
Would
was,
you have been interested in studying that water if you'd had the opportunity, if it wasn't contaminated, or is it just not your kind of area of
yeah.
expertise?
No, that would be interesting to look at that water to compare it with the accretion ice above, that's already been studied. But yeah.
Okay, lovely. Well, thank you so much for talking to me today. It's been really interesting, and I really appreciate you taking the time.
Well, thanks for your interest. Yeah.
Amazing. So yeah, so I yeah, we'll do kind of a bit more research and kind of write it up. But yeah, thank you very much, and I hope you have a lovely rest of the day.
Okay. You too.
Fantastic. Take care.
Now, as far as the recording,
Oh yeah.
what do we do with that?
Yeah, if you can stop the recording, perhaps it'll
Yep.
convert, and then if you can maybe